going to be extremely basic stuff, and I will move fairly fast unless you want me to stop and explain something. So, if you want, please stop me, and the aim is to get to a biological example at the end of the day, whatever the picture was. We will see if not today, then next day. Right. So at the very basic is, so if you, let us say you have some experiment, where you do an experiment multiple times. Let us say you do an experiment n times, right, and you measure the outcomes of that experiment, and some outcome you define as whatever you're calling as your desired outcome or success, let us say success, okay, whatever that outcome is. So, you perform an experiment n times, out of that, and let's say you get ns number of successes, and then you say that, well, then the probability of success is simply ns over n, except that there is a asymptotic behavior. So, it is in the limit that you do an infinite number of trials, right. You do this experiment again and again and again, provided you have enough statistics. This number of successes by the total number of trials gives you your probability. This is in some sense what is called as the objective measure of probability. In some sense, what is known as the objective measure of probability, where you do some experiment and then you find out what is the answer. You could start the other way, which is the subjective way, which is you assign a probability and then you do experiments and then you revise your estimates based on whatever is the outcome of the experiments. That would be the subjective or the Bayesian approach to probability. And this is basic stuff. Now, when we are talking of probability, we can talk either of discrete outcomes. So, we can talk either of discrete outcomes, or let us say discrete random variables, or of continuous outcomes or continuous random variables. Right. So, for example, let me say, to take this example that we were talking about the last day, I have my DNA, then this transcribes into RNA, and then this produces some protein. I ask at some point, what is the probability that inside a cell I have 5 copies of a protein, or 6 copies of a protein, or 10 copies of a protein. So, the outcomes there are countable, right, and discrete, and therefore, the probabilities that would be associated with these outcomes are discrete. So, therefore, these are discrete variables, and the probabilities associated with these variables will define a discrete random variable. So, in some sense, you have some set of outcomes. Let us say J is, however, depends on what you are doing, and then you have a probability associated with each outcome, right, P of Aj. So, this set and their associated probabilities together define for you your discrete random variable, and just like with any probabilities, this satisfies certain conditions. So, for example, these lie between 0 and 1, right. A probability of 0 means that elementary event is impossible. It's never possible to observe that event. A probability of 1 means that elementary event is certain. You are guaranteed to observe that event. And in general, of course, you must have normalization. So, if you sum over P of Aj, sum over all values of J, you must get 1. So, that is your normalization. So, this is my discrete random variable. On the other hand, I could have outcomes which are continuous. So, when I perform an experiment, for example, again to go back to this example of DNA, I have my DNA chain, It's a very bad double helix, but this is a DNA double helix, and I have this ribosome or whatever RNA polymerase protein that is scanning along this DNA, searching for the target site where it can start the transcription of a gene, right. So, it is moving along this DNA backbone, and I asked where at some instant of time is this what is the position of this RNA polymerase molecule. So, in that sense that is a position, and the position can take any value along this DNA backbone. So, this is the set of outcomes in this case is continuous. So, in principle in this case of course the length of the DNA, but in general any continuous outcome between minus infinity to plus infinity, right. And again you can, in this case what you can define is a cumulative distribution function. You can define a cumulative distribution function that's it, capital P of x, which tells you the probability of the random variable this outcome being between minus infinity and x. Okay. Okay. So, this is the probability that, so this is the probability that your outcome lies between minus infinity to x, right. And therefore, if I think about this cumulative probability distribution and I were to plot it, this P of x versus x, at minus infinity of course it is 0, right, because you are starting from minus infinity, and at plus infinity it has to be 1 because you have to have some value of the random variable in between, which means it will look something like this, where asymptotically it will reach the value 1. It could be anything, I mean it could be something like this, it could be something so. Could it be something like this? No. So, it could be something like this and so on, but in general it is a monotonically increasing function, right, because you are keeping on adding probabilities. It starts off at 0 and it reaches 1 at as you at the upper bound, wherever that upper bound of your range of variable is. From here you can define what is the probability density function. You can define what is the probability density function PDF, right, small p of x, let us say, and this is just the derivative of this cumulative probability capital P, and what it tells you is, so this small p of x dx is essentially the probability of an outcome that lies between x and x plus dx, right. So, what is the probability that you perform this experiment, you get an outcome between x and x plus dx, that is your px dx. Because this is the derivative, there is no restriction on the magnitude of this, well it has to be positive because it is a monotonically increasing function, but that is essentially all you can say, that px is greater than 0. Right. It can be as large as you want depending on how steeply or what is the behavior of this cumulative probability. If it rises very steeply, then in this region you will have a very high value of p of x because the slope will be very high. Okay. So, this is essentially px, and of course, you have to be normalized, which means that px dx must be equal to 1. Right. That is your normalization condition. This is of course for one variable. You can now extend this to probabilities for many variables. Right. So, again and what you get are the joint probability distributions. So, let us say if I am thinking of discrete variables, let us say I have some set of elementary events A, another set of elementary events B. In the trivial sort of example, you have 2 proteins, you ask how many copies of this protein A, how many copies of that protein B, and you will have some joint probability distribution P of A comma B. And again, these should obey the same sort of conditions. It has to be bounded between 0 and 1, and if you normalize them, so you summed over all outcomes of A and all outcomes of B, these would be normalized, right. Similarly, you could have a set of continuous outcomes x, which is some x1, x2, xn, all of which are continuous variables. Again, in this sort of an example, because you know that there are multiple genes, there are multiple RNA polymerases typically that are searching for transcription start sites, and each of those, the positions of each of those could be a random variable, and then you will have some sort of probability density function which depends on this vector x, which has all of these components, right. And again, in this case, you will have some normalization. So dx1 all the way up to dxn, p of x1, x2 dot xn, this is equal to 1. So you will have the appropriate normalization. By the way, talking about this reminded me, somebody was asking me about start sites and what happens when there is another start codon in the middle. I don't remember who it was, but it can happen, and then that methionine just gets absorbed into the protein structure in that sense. Okay. So if you have a transcription start site which starts with the start codon, the protein the transcription starts. If you have another AUG in the middle, then that just gets that becomes part of the protein structure. So it continues until it hits the stop codon. However, there are issues apparently. So sometimes, because you have these start codons in the middle, sometimes you get transcription starting instead of from the proper place, it starts from here, and this is called leaky transcription. So errors can happen in some sense. Okay. But yeah, in general, it's possible to have the start codon in the middle of the sequence as well. So that's my many variables. Once I have many variables, what you can, so these are the joint probability distributions. So when you have many variables, what you define are the joint probabilities. Right. And then from the joint probability distributions, you can extract information about any one of the random variables. By the way, so if you have probabilities of many variables, if these random variables are independent, so for example, I have something like this, p of x1, x2, xn. If these are independent, if all of these random variables are independent, then this factorizes, then and only then, it factorizes into a product of probabilities I equal to 1 to n pi of xi. Right. So if your random variables are independent, the joint distribution is simply a product of the individual distributions. Like I was saying, so if you have these joint distributions, you can extract information about the individual variables. So that's called the marginal or the unconditional probabilities. So let us say I have a joint probability distribution, P of A comma B, right. I want to know the probability distribution of this variable A. Okay, so I want to know P A of A. What would I do? What would I do? Sum over all the instances of B for this value of A, right. Or similarly, if I wanted to get PB of B, I would sum over all instances of A. Similarly, in the continuous case, you integrate out over the variables that you don't need. So for example, I have some x1 to xn. Let us say I want to know the joint distribution of only a subset of those, xk, right, x1 to xk. Then what I would do is that I would integrate out dx k plus 1 all the way up to dxn of this full probability distribution, right. and this would give me the unconditional probability for this subset of random variables, independent of the values of all of these other variables. So, that is my marginal or my unconditional probabilities. Counterpart to this, we have the conditional probabilities, where you ask for a probability that is conditional on some value of the other variable. So, you have conditional probabilities, where instead of this joint distribution, what you ask is what is the probability of an outcome A of one of these random variables, given that you know what is the value of the second variable, right. So, P of A given a value of B. And again, the way to do that is to write down the joint distribution P of A comma B. So, this is the joint probability distribution P of A comma B is the probability of A given B times this marginal probability of B. So, PB of B, right. So, what is the probability of A given that B has happened times the probability of B itself happening, or even in the reverse way both is possible. So, P of B given A times the marginal probability of A, right. So, you can write the joint distribution in terms of this conditional times the marginals, and then you can write these conditional probabilities P of A given B, let us say, as P of A comma B times PB of B, but PB of B is nothing but this sum over A. So, let me write this P of A comma B, let me rewrite this in terms of this. So, this is P of B given A, P A of A divided by this PB, which is sum over A P of A comma B, right. And I can again substitute for this A comma B from over here. So, P of A given B is P of B given A, P A of A divided by summation of A of P A comma B, and there again I substitute this, so P of B given A, P A of A, right. And this gives us Bayes' theorem. The conditional probability of A given B in terms of the reverse conditionals, the probability of B given A. Right. So, this is Bayes, and again we will come back and look. So, Bayes is used everywhere, we will come back and look at the applications of Bayes in certain biological contexts as well. All right. So, that is conditional probabilities. Then once you have probability distributions or probabilities, you look at central measures. So, central moments of the probability distribution. So, essentially mean, variance, moments, right. And again, if you have some probability distribution P of x, the mean of course is xi. If it is a discrete probability distribution, it is xi P of xi sum over I, or if it is continuous, then dx x px. That is my mean. If you have some function of these random variables, some f of x, then the mean of f is f of xi or f of x, p of x. In general, you can calculate the nth moment. So, x to the power of n, which is dx x to the power of n p of x, right. What is often done is to calculate the central moments. So, instead of x to the power of n, you subtract the mean x minus x average, and then you take the nth moment of that, right. That is the nth central moment. And the second central moment, so if you do x minus x average whole square, this you can show is nothing but x square average minus x average whole square, which is the variance, right. And in general, given a probability distribution, you can find out the characteristic function, which will generate for you moments of all orders, right. So you can calculate, you can define the characteristic function of a probability distribution. Let the characteristic function, right. So p tilde of k, which is nothing but the Fourier transform. So e to the power of minus I k x average, right, which means that it is nothing but dx e to the power of minus I k x p of x, right. And why this generates all the moments is to see that you can simply expand this exponential. So integral dx sum over n equal to 0 to infinity minus I k x to the power of n by n factorial p of x, right, which means if I pull this summation outside, n equal to 0 to infinity minus I k to the power of n by n factorial, and then if I keep all terms related to x, so dx x to the power of n p of x, and this is nothing but your nth moment, which means that in this series form minus I k to the power of n by n factorial, this is nothing but the nth moment. So knowing a probability distribution p of x is equivalent to knowing all of the moments of the distribution. So you can either know the function, you can either specify the functional form, or you can specify all of the moments of the distribution. Either is equivalent because you can go from one to the other. If you know this, you know p tilde of k, you do an inverse Fourier transform, you get p of x. Okay. And then of course, often people will talk of cumulance, which is the log of this p tilde of k, which is another measure of these central tendencies. But anyway, whatever works. I mean, you can either talk in terms of the moments or the central moments or the cumulants. These are all alternate descriptions of the probability distribution. And knowing any set is equivalent to knowing the full distribution itself. Okay. Good. This is as far as basic probability goes. Right. All right. So let's try to think of one example. Again, a very familiar example, which is that of the Poisson process, because that's something that we'll see again and again. In various biological contexts. Okay. Let's talk about the Poisson process. What defines a Poisson process? Since most of you I think know what is a Poisson process. What defines a Poisson process? That's an example of a Poisson process, definitely. What is a Poisson process? By the way, let's since there's a small class, I'd actually like to know all of your names. So whenever I ask, please can you just Abhishek, PhD physics. Okay, Abhishek. So I'll have to do this 2-3 days before I remember names. Okay, so excuse me. So what defines a Poisson process? Anyone? What is the defining characteristic, or what are the defining characteristics of Poisson process? Yes, Shujit? This is elementary, right? Discrete events in a continuous time span. But what governs these discrete events? The probability is, what this says is that each, essentially what a Poisson process is that each of these events that are happening in this continuous time, each event is independent of the other. Okay, of all other events. So you have a Poisson process where you have discrete events happening at discrete times. Each of these events are independent of all other events. And secondly, events occur with some rate. Events occur with some rate, right? And let me call that rate R of t. In general, perhaps when we generally do Poisson process, it's a constant rate R, but in principle that rate could be a function of time. There is no problem with that. So as long as it's some defined function of time, it's some R of t. Okay. If it's R, if it's some constant, so if R of t is some constant, let's say R bar, then this is called a homogeneous Poisson process. Homogeneous Poisson. And therefore, R of t basically is that so I perform my experiment many times. At some time t, in some time interval, let's say between t and t plus delta t, I ask what is the rate at which an elementary event happens in this time window. And I do this experiment many times. I say that I find the average rate, and that average rate is what is this R of t. So these are the two sort of central defining characteristics of a Poisson process, that each event is independent and these events occur with some defined rate R of t. So, and what do we want to say? So, what do we want to calculate? So, essentially, so this is my event that is happening in this time axis. So, this axis is time. Let us say an event has happened at some time t1, another event has happened at some time t2, third event has happened at some time t3, okay. Fourth event has happened at some time t4, and so on. And I want to know the probability of this composite process, right. So, one event at t1, another event at t2, another event at t3, and so on and so forth, and a final event at, let's say, some tn, right. So, I want to know what is the statistics of these elementary events happening at these time intervals. Okay. So, what I want to calculate is this probability of this sequence of events, right, t1, t2, t3, t4, tn, given some rate R of, since I am using t, let me just call it R of tau. Okay. So, I want to know the probability of this sequence of events, where each event is independent and they occur with this rate R of tau. And I could imagine that what will this depend on? So, essentially I have this elementary events happening at t1, which means that this probability I can sort of assume is going to depend on this rate at time t1, where this first event happens, times the rate at time t2 when the second event happens, and so on, times the rate at time tn when this nth event happens, right. So, it must be proportional to the product of these rates evaluated at the times of the elementary events t1, t2 all the way up to tn, right. Why? Because these events are independent, and we said that if events are independent, the erase it, but the total joint probability is a product of the individual probabilities, and the individual probabilities of event happening at t1, at t2, and so on and so forth is this. It's just a dummy time variable, because I am writing t, I will be, so instead of t I just write tau, it's a time index. but it is not just this, right, because this tells me that there is an event that is happening at time t1, another event that is happening at time t2 and so on. But I do not just want this, I want that no event is happening at times other than this, right. So, I have to account for that probability as well. It is not just events happening here, here and here, but the fact that no event has happened anywhere else in this along this time axis, right. So, in general the probability that an event has happened at time t around some time interval delta tau is this, right, the rate at t times the window of time delta t, right. This tells me that an event has happened between t and t plus delta t, right. Then the probability that no event has happened in this time is 1 minus p of t, right. So, this is no event has happened. Then to calculate this probability, I need to have the probability that no event has happened at any time other than these specified times, right, which means that, let me define this function. So, let me define this function. So, f it is a product of these things 1 minus p of t something. So, here the final event, let me call it as t capital N, and this is some t small n, okay, where the product is over all n's not equal to, so for product is over all t n's not equal to t i's, right. So, events happen at t1, t2, t3. So, the probability that no event has happened at any other time is this factor for each time product over all times which are not part of this elementary set that I have started with, okay, t1, t2 and so on. Okay. So, that is this factor, which means that if I were to now write this, so it is not just this, but it is also this, right, because this is the probability that no event has happened, right. So, if I wanted, so since I was writing this thing, so probability of Ti given some rate r of tau is this factor. So, it is product of n not equal to i's 1 minus p of t n, right, times this factor, the probability that the event has occurred at these times. So, product over I equal to 1 to n r of Ti, let me put a delta tau here and then let me just put a delta tau to the power of n here. Since there is a probability density and therefore, this whole thing is the probability that I have this events happening. And this, and I have this overcounting factor. So, how do I label these events 1 to n, which has this factor of 1 over n factorial. So, if I have n events, I have this factor of n factorial due to the labeling, and this p of t n is nothing but r of t n times delta tau. So, this is nothing but r of t n times delta tau. So, this is my elementary probability, okay, of this sequence of events happening at these times. Now, of course, these types of products are difficult to do. So, what you can do is that you can do this product over all n's instead of having this n not equal to I, you can do this product over all n's, okay, right. What that means, I have these extra factors corresponding to every I, which means that I can just divide this by 1 minus r Tn delta tau, okay. And this becomes an unrestricted product, because these extra terms I have just absorbed into that over there. Fine? No, lost? Kahan pe? No, tell me if you are lost, right? Because here, up till here, did you understand that this was n not equal to I? Because these are the times when the event is not occurring. Now, if I have removed this sum, what means that I am doing these extra products where these n's take these values I. So, 1 minus r T1 delta T, which means I am doing this extra. So, I divide by that, and those terms I just take inside this. So, then this is what I. Yes, anyone else? This one? This one? See, I had product of n not equal to I, 1 minus r Tn delta T, delta tau, okay. Which means that, so let me think about, if I just discretize my time axis, let us say here I have, I don't know, let me call this something, between T1 and T2 I have all of these other times, right, where the events are not occurring. So, let me call that PQ S, okay. So, S1, S2, S3 all the way Sn, right. So, what this means is that when I am doing these products, I am doing 1 minus r S1 delta tau, 1 minus r S2 delta tau, and so on, all the way up to 1 minus r Sn delta tau, okay, because at each of these times the event is not occurring. Now, I do one additional one, 1 minus r T2 delta tau, where T2 is one of these times where the event has occurred, okay. So, I do this product as well at this time T2, but this originally was not there, because, right, which means that I multiply and divide by 1 minus r T2 delta tau. So, now if this denominators I have just taken to inside, and then in the product is an unrestricted product over all possible times. RTI, sorry, this is an RTI. It's to do with, I have an event occurring at time T1, another event occurring at time T2, another event occurring at time T3, which, how do I label these events if these are distinguishable? So, which one do I call event 1 and so on. So, that is the originally Gibbs correction, overcounting correction, okay. This is the same thing, it's a labeling thing, right. So, I have an event occurring at time T1, another event occurring at time T2. I could have said that event 1 is occurring at time T1 and event 2 is occurring at time T2, or event T2 is occurring at, so this is label overcounting due to the labeling. Okay. This is the extra step. So this is what I have. Now I can evaluate each of these up to order delta tau. I just want to evaluate up to lowest order. So let us say I look at this term, product of n 1 minus r of Tn delta tau, right. This I can write as an exponential of a logarithm. So this I can write as an exponential of a summation over n log of 1 minus r Tn delta tau, right. This product in the exponential becomes a summation. And now I look at this logarithm, this is log of 1 minus something, and these delta taus are very small time intervals, right, because I am looking at a time T1 and some small time interval delta tau around this times. Okay. So, this delta tau is a vanishingly small time interval. So, we will look at delta tau going to 0 limit, which is why we will keep only terms of lowest order in delta tau. So, log of 1 minus x is so therefore, what I get is exponential of minus r of T n delta tau. There is a summation outside, right, and then plus a half of r square of T n delta tau whole square and so on, and there is a sum over n. So, just do a log of 1 minus x. When del log of 1 minus x, yes. All are minus. Okay, is that correct? Okay. And now, in the limit that delta tau goes to 0, this summation becomes an integral. So, essentially what you have over here is an exponential of minus integral dt r of t, right, and then terms which are of order delta tau, which I will draw. So, plus higher order. So, from this term to lowest order, I get exponential of minus dt r of t. Similarly, from this term r of t delta tau product over n to lowest order again, what I will get is so I have a product of I equal to 1 to n r of Ti delta tau by 1 minus r of Ti delta tau. Again, to lowest order, this is nothing but delta tau to the power of n product over I equal to 1 to n r of Ti, right, and then you will get terms which are 1 minus delta tau times something, which is actually again this product, n r of Ti and higher order, so on, right. If I do 1 by 1 minus x, right, expand this systematically in orders of delta tau, the lowest order term is delta tau to the power of n product of I r of Ti, and then 1 minus higher orders of delta tau, which again I will neglect exactly like I have neglected there. So, then if I bring everything together, what I get is for this joint probability distribution, P of events happening at T1, T2 all the way up to Tn, given this rate r of tau is 1 over n factorial, the Gibbs factor, exponential of minus dt r of t. This goes from 0 to t, whatever is the time interval, the final time t that you start your clock at some time 0, you end your clock at some time t, n events have happened in that time at these times. So, that is that 0 to capital T, and then you have this this product over I equal to 1 to n r of Ti, right. This delta tau to the power of n and this delta tau to the power of n go away. So, this is the probability density of having this set of events at these set of times, right. Okay. So, this is the basic joint probability distribution of a general random process, in a homogeneous random process where the rate is a function of time. There is a joint probability distribution of these events happening at these sequence of times Ti's. Okay. Now, of course, I should check that. Sorry. here, here. So, I have r of T I delta tau by 1 minus r T I delta tau, right. So, because delta tau is small, okay, this is like something like 1 by 1 minus x in the limit that x is small. So, this is like r T I delta tau into 1 plus r T I delta tau plus r square T I delta tau square, correct. And then I am just keeping this lowest order term. Okay. Anyone else? Okay. So, I should check that this is normalized, right. So, or rather you can check that this is normalized as an assignment, but what is the statement of the normalization? So, I have this thing, right. Or let me just write it out like this, this sequence of times T1, T2 all the way up to Tn given a rate, a time dependent rate r of tau, right. So, this is the joint probability distribution that I have now calculated. What is this? What is the mathematical expression for normalization of this? What do I integrate over, or what do I sum over? So, something about this probability has to be equal to 1, because, but what is that condition? All the Ti's, because the times are continuous. So, dt1, dt2, dtn each from 0 to t. Yes, anything else? Is this correct? Anything else? Hillel, anything else? Say that once more, sorry I couldn't. This tau or this capital T, which this one is the joint probability, that I have events happening at T1 and at T2. This is not the conditional, this is the joint. Okay. Correct. So, this event is essentially, so this is my experiment. I am starting my experiment at time 0, I am finishing my observation at time capital T. So, this is some sense a parameter, but so that is why all those integrations go up to T, but you want to do something with this T, capital T to the power of where? Here? T to the power of n? Why? Sorry, I forgot your name again. Pavitra. Okay. Why Pavitra? But if these probabilities are normalized, essentially over each integral this would give me 1. So, probability dt from 0 to t would give me a 1 if that was a certain event. But what you can think about is what is this n? There will be a sum over n, because there is no guarantee that how many events you are going to observe, right. So, therefore, in this time t you can have no event, you can have an infinite number of events, right. So, if you are normalizing this probability distribution, you have to do this summation as well, and then you are guaranteed that it will give you 1, right. So, this tells me that what if there are n events, what is the probability that it happens at such and such times, then you integrate over the times that these n events can happen, but then you also have to sum over the fact that this n can be anything, you are just doing your experiment from 0 to t, maybe 1 event happens, maybe 10 event happens. So, you have to do this summation, okay. So, this is the normalization condition, sum over n, integral over all of these t's, and you can take this expression and check that this is actually normalized. Okay. What we want to do in the Pursat process is to calculate the probability that some n number of events has happened, right. So, what we want to do is to calculate the probability that some n number of events has happened. So, I want to calculate what is the probability that n events has happened. So, what do? I pick out these terms which have only n events, only n times in this probability joint probability distribution, and then I integrate over all of these times, because these n events could have happened at any time, right. That is the probability of n events happening. Okay. So, that is the first part. So, dt1 to dtn 0 to t 0 to t, probability of these ti's where I have n events happening. So, these ti's are now n events only, again given some rate r of t. And I know the expression for this this thing. So, this is nothing but, so integral dt1 all the way up to dtn 0 to t 0 to t 1 by n factorial exponential of minus dt r of t 0 to capital T product over I equal to 1 to n r of Ti, right. So, just this expression is what I have written there. Now, my integration variables are T1 to Tn. This term over here has no T1 to Tn, so I pull it out. This n factorial has no T1 to Tn, so I pull that out. So, what I am left with is these dt1 to dtn, and then these products of I equal to 1 to 10 r of ti's, and these integrals are all independent because these elementary events are all independent, that is my Poisson process. So, essentially what do I have? I have a 1 over n factorial exponential of minus dt r of t from 0 to t. So, I have just pulled that out. Now, inside what I have is just these dt prime r of t primes, where this t prime is any one of these t1 to tn, and I have the same integral n times to the power of n, right, because each elementary event is independent. So, essentially if I now this integral if I call as something, so let me call it q dt r of t 0 to t, then essentially what I have over here is 1 by n factorial e to the power of minus q to the power of n, right. This is the probability of having n events. In the normal Poisson distribution, the one you are familiar with, I hope, what comes in place of this q? RT, right. So, that is for this homogeneous process. If R was not a function of time, it would just pull out, you would get RT, which is nothing but the mean of the Poisson, right, the mean of the Poisson process. So, here also in this general Poisson process where this rate is a function of time, one can calculate the mean. So, here is this P of n, I can calculate the mean, which is of course NPN. So, I can calculate the mean of the Poisson process, average n, which is what? Which is sum over n equal to 0 to infinity n times P of n, right. P of n is over there. So, this is sum over n equal to 0 to infinity 1 by n factorial e to the power of minus q to the power of n times n, right. This is sum over n, this q is just something dtr integral dt rt. So, I can just pull that out. So, this is e to the power of minus q sum over n equal to 0 to infinity q to the power of n times n by n factorial, which I can write as e to the power of minus q, this is the standard trick, q del q of sum over n equal to 0 to infinity q to the power of n by n factorial, right. Because if I take a del q, I will get a n times q to the power of n minus 1. I multiply by a q, that would give me a n times q to the power of n, and this n factorial just stays in the bottom. but this is nothing but what? Q to the power of n by n factorial is just e to the power of q, e to the power of plus q. So, essentially what I get is e to the power of minus q, q times del del q of e to the power of plus q, which is nothing but e to the power of q, which means the mean in this case is simply q. So, it's just simply the time integration, the time integrated rate r of t. So, this is the standard generalization of this homogeneous Poisson. Instead of r t, you get this integral dt r of t. So, if you have some time varying rates. One could also ask that, so this then gives you the standard identification in terms of this average n. This gives you the standard identification of the Poisson process in terms of its mean, which is that the probability of observing n events is e to the power of minus average n to the power of n by n factorial. Right. You could also ask that what is the probability that in this time 0 to t I have observed no event. So, you could just set n equal to 0. So, the probability that no event occurs between 0 to t is simply p of 0, right. And so, p of 0. So, essentially I would get 0 factorial is 1, something to the power of 0 is 1. So, I would just get this. So, this is nothing but exponential of minus q. So, minus dt r t. The probability of observing no event is just exponential of minus q. Again, you can do various things. You can calculate the variance and show that it is equal to the mean, just like you would expect for a Poisson process. Another thing that you can calculate is the interval between events. Okay, that's another thing that maybe let's calculate that, because I'll try to use it next day or something. All right, this is fine. You've got your Poisson for general Poisson for a time dependent rate. In the case when r of t is constant, all the standard terms come out. You could ask about the interval between events. Okay, so you could ask about the probability distribution. What is the probability distribution for intervals So, one way to do that is so we can start by asking that what is the probability that I observe 0 events in a time interval between t and t plus tau, let us say. Okay. So, what is the probability that I observe 0 events between t and t plus tau? I know what is the probability to observe 0 events between these two times. So, I can simply put t here, t plus tau over here. That would be my probability of observing 0 events, right, in this time. So, this would be exponential of minus t to t plus tau dt r of t, correct. I have just taken this and changed the limits of the integral appropriately between t to t plus tau. Now, what I am looking is the interval between events. So, it is not just no events have happened between t and t plus tau, but rather the fact that an event has happened at t, then no event has happened between t and t plus tau, and then another event has happened at t plus tau. That gives me the interval, right. So, therefore, if I write this, what is the probability that I have an event at t, an event at t plus tau and no event in between? That is what? That is simply the density, if I write. So, that is simply r of t. This gives you the probability that an event happens at t. Then nothing happens in between, which means I have an exponential of minus dt r of t between t to t plus tau, and then another event happens at tau, which means I have a r of t plus tau. And then what I want is this average probability to observe the two events separated by this interval tau, which means I want this average probability. So, I take an average over this, okay, over all times t, or essentially over variations over the rate r, right, such that the event interval remains tau, but the time can be anything, right. So, that gives me this average probability. Here, so here remember I have calculated what is the because I know the probability of n events happening, I have calculated what is the probability that no event happens, and this no event happens in a time interval over experiment 0 to t. But now my experiment is between t and t plus tau, which means that my lower limit is t and my upper limit is t. This expression over here. Here? No. So, this is just saying that, so what is this saying? This is saying that I have a time t, I have a time t plus tau. This thing over here is the probability that no event has happened here, but it could be that no event has continued to happen over here or over there. The lower one is not this, but event here, nothing here, and then event here. So, that is the difference. So, that is why I have a event at t and an event at t plus tau sandwiched between this no event. This is just, it could have continued that interval. Good. So, this is then the average sort of probability for observing two events separated by a time interval tau. This in short is written by P2 of tau, probability of having two events separated by an interval tau. And then if I want the probability density at intervals of this intervals, so if I want this, the probability density of intervals between events, what I want is essentially the conditional probability that given that an event has happened at t, what is the probability that an event will happen at the next event will happen at t plus tau, right. So, this conditional probability is this joint probability, which is this. This is the joint probability that I have an event at t, I have no events in between, and another event at t plus tau, right. So, let me just call this P2 of tau divided by the probability that an event actually happens at t, which is essentially this r of t, which is essentially this r of t. And in general, of course, depending on what the rate is and so on, you can find this out. But you can see that, for example, if I had a homogeneous Poisson process, so if my rate was constant, then I would just have an r square over here and an e to the power of minus r tau, because this would be constant, this would just be tau divided by r. So, this would be r e to the power of minus r tau. Right. So, this sort of a distribution, the probability distribution between events given by r e to the power of minus r tau, right. This is a signature of a Poisson process and essentially says that again it goes back to the fact that the underlying elementary events are all independent, they are uncorrelated in time, there is no memory of the process. So, if at some time t some event occurs, then the process has no memory once it has passed this time. The next event is equally likely at any time event, and this is a signature of that, this R, and you will see this sort of distribution, interval distributions cropping up again and again in various biological contexts. Okay? Yes? For P2 tau, yes? There should be a 1 by 2 factorial. Why is that? No, I mean there may be, but why? No, so here I already have a time order established, that I have this event which is happening at t, followed by an interval, followed by an event which is happening at this r t plus tau. Right. So, I have already put the label that this is my first event which happens at time t, and then the second event which is happening at time t plus tau. So, there is no A for counting anymore, any counting ability. Alright. So, to give a nice example of this, this example is, so now that we have talked about Poisson process for a bit, which one? This is the interval distribution, that what is the probability distribution of having an interval tau between two events. This is the joint probability distribution, that I have an event at t and another event at t plus tau, and then I want the interval. So, I divide by the probability distribution that the event happens at t itself. So, then I get the, so on a conditional probability. Which here? How do you mean there is a degeneracy? Why again, can you explain? This is some dummy index, of course, I mean this is some t prime, and then I do a dt, right. So, one event happening at t, an interval of tau, and then another event over all possible times. So, no, so this, so I have this interval 0 to t, let us say. This probability is only concerned about one part of this interval, which says that there is an event here, no event here, and then another event here. There can be anything outside. Does not talk about that, because it's not talking about how many events. It's just talking about this interval. It's just talking about the interval between these two events, and then it's taking this window and translating it over this whole process to get what is this average probability of this interval tau. Why is this average taken? Because right now there is a t. In principle, I just want to get this interval tau, right, independent of, so I average over all times. So, this is in some sense, telling me that, so P2 tau is telling me that I have an event at some time, and then I have had a gap of time tau before the next event happens. Okay, so two things. So, an event has happened, and or rather plus interval of tau, right, and what I want is essentially this conditional probability. One second. So, this is the, so this is two things, event has happened plus an interval of tau. This is the product of the conditional, that an interval of tau, given that event has happened, times the probability of that event happening, right. And so, this P of tau in the last line is this conditional probability, but I have divided by the probability of this event happening. Mathematically, I mean it means this conditional probability over here. Let me see if I can say this better. So, again, so essentially perhaps I am saying the same thing. So, essentially what I am saying, if I am doing an experiment, I see some event and I ask that now what is the probability that given that I have seen this event, I will have to wait for a time tau until the next event happens. That is actually, this is the difference between the joint and the conditionals, right. So, the P2 is saying that yes, an event has happened here and the next event happens at t plus tau. So, this is the joint probability. This is the conditional saying that given the event has happened, what is the probability that I now have to wait an interval. So, these are connected because you both have these two events, that something has happened at t and the next is happening at t plus tau. It is how you interpret this conditional probabilities versus this joint probability distributions. But essentially, just to give you a backdrop, so essentially this is a very beautiful example from Bill Bialik's book, this Biophysics Searching for Principles. So you can look up that book if you want to. I'll just discuss it briefly, but it goes back to this experiment by this experiments or these calculations by Lorentz, and then these experiments by this Dutch group, I think whose names I have now forgotten, but I will tell you next day. So they said that if I am sitting in a very dark room, right, and you flash a light source, at some point if your intensity is very low, right, if it is a very bright light, I see that light of course. If your intensity is very low, then whether I see that tiny flash of light or not is a probabilistic event, right, because that light source is, so firstly the light is not a continuous wave, because by that time it was already the quantization of light had been, that theory was more or less accepted. So people knew that light was travelling in quanta, and this emission was a probabilistic event, right. Some quanta of light were emerging with some average intensity, right, and then that quanta of light had to hit your eye, and that detection is also a probabilistic event, right. So it hits you the retina of your eye, there are rod cells in my retina, that leads to some chemical changes, that gets converted to an electrical signal into a brain, and then you say that whether the light has been flashed or not, right. So what Lorentz asked is that, you know, what is the sort of roughly, if I do a back of the envelope calculation, what is the number of photons or what is the number of quanta of light that I need in order for me to say that there is some signal. So what is the minimum number of quanta of light that is needed? And Lorentz did this, I mean, you know, you take light in the visible range and so on, and you take Planck's constant, and he came up with a number that this was roughly around 100 photons. Okay. So then he asked that, well, you know, this is of course a very hand waving calculation. Can I actually estimate this number with a little more certainty, that what is the minimum number of photons that I need to strike my retina in order for me to say that there is a light signal or not. Okay. And in fact, the interest in some sense is to know that if I have these cells in my eye, what is the sensitivity of these cells? If a single photon comes and falls on one of these retina cells, is that enough for me to sense? Okay. So that is what he asked, and that is what these people did an experiment in the 1940s to find out that what is this limit of light detection, okay, purely from the approach of counting photons. And photon emission is a Poisson process, because it is each photon is being emitted in an uncorrelated way. So that is why we will use Poisson statistics, or rather they use Poisson statistics to estimate what is this minimum number. Okay. So I'll talk about that next day on Thursday. Think about this. If there are any further questions, you know, let me know. I'm happy to discuss. No, as far as I know. I mean, there are things that the frequency of light or various other biochemical changes they matter, but not in this sensitivity sense. So that number is independent of that frequency. So they actually in the experiments, I think they used light. I will check. I think they used lights of certain frequencies, and they repeated it using different frequencies to get what was this threshold number of photons that you need. Okay. So yeah, so I'll talk about this in a little more detail on Thursday.